Hi everyone, thanks for coming to my virtual presentation. My name is Olivia Rose Hamilton and I'll be talking about female túngara frogs and their memory. As we're all well aware, the animal kingdom is diverse, and so much of that diversity is a result of the way animals communicate. Whether it be the aposematic coloring of a poison frog, the song of a male eastern bluebird, the pheromone and chemical cues of a big cat, or even as something as intricate as the nest of a male pufferfish trying to attract a mate. Often when animals communicate, they do so in more than one sensory modality to form a multimodal signal. Multimodal signals are especially prevalent in the context of mate choice, such as in male butterflies who use a combination of both visual and chemical cues to attract a mate, or in wolf spiders who use a visual and seismic cue. And maybe a little bit more relevant to my talk, male frogs who use visual, seismic, and acoustic components to attract a female. But why do females pay attention to multimodal signals in the first place? Well, decades of research have provided evidence that multimodal signals may help inform females' decisions. One of the leading hypotheses within the field says that a multimodal signal may provide a receiver, in this case a female, with more information from a signaler, usually a male. And as one of the classic papers by Rowe in 1999 stated, multimodal signals may help a female detect, discriminate, and remember a male, which the memory aspect of that I'm going to be focusing on for my talk. Let me introduce you to my study organism, the túngara frog, Physalaemus or Engystomops pustulosus. This species is native everywhere from Mexico to South America, including Panama where I study them. Males of this species gather in large leks with sometimes more than 100 individuals to call for females during the rainy season. The loud choruses at ponds can stop, however, if something like a predator passes by or even if I walk by with my headlamp. These pauses, referred to as intercourse intervals, last on average for 25 seconds. Like I said before, the multimodal signal of the túngara frog is comprised of an acoustic signal coming from the call, a visual cue of the male's vocal sac inflating, and a seismic component as ripples come off from their body when they inflate their vocal sac, all of which we can see with this little guy. We know that the acoustic aspect of their signal takes primacy over the other two components, and so for my research I focused on the acoustic and visual aspects of their multimodal signal. In our lab, we're able to recreate this multimodal signal with the use of a robo-frog, or a robotic frog. Our 3D printed, hand-painted frog models have a silicone vocal sac that inflates in time with the speaker, while another speaker remains unimodal with just a call. We know that female túngara frogs have a preference for the multimodal signal over the unimodal signal, as we can see in this video. In the previous videos, we've only heard one type of call, but male túngaras have two basic call types, a more simple whine call, and a more complex call that we've heard before, a whine chuck. The extra note at the end of the whine chuck, chuck, is very attractive to females, and males are actually able to add up to 7 chucks at the end of a whine. What's interesting about this is that when they increased the complexity of a túngara call with chucks, Akre and Ryan were able to show that it made the call more memorable to females. Since then, not much research has been done in the field of memory and multimodality in the context of mate choice, with one exception being a paper that was published by Zhu et al. Last year, where they used female serrat-legged tree frogs from China and showed that a multimodal signal that they created with a video playback was able to instantiate the memory of females in that species. What we have to remember is that we're comparing a tree frog to a túngara frog, and they have different life histories. Túngaras are going to be on the ground in the water making a choice, which typically isn't going to be the case for a tree frog. Still, both species are not going to be living in a homogeneous environment. Their environment, as all of nature is going to be constantly changing, whether that be throughout the night or even from a female's perspective as she moves around in search of a mate. Túngara frogs are really small. 2 grams is a chunky gravid female. She's 25 millimeters long. So if she can see a calling male from one angle, she might hop behind a clod of dirt and that male is no longer visually accessible to her. If her environment has visual obstructions, does she rely on memory to make a choice? So that leads me to my ultimate research question. Does the visual component, i.e. the vocal sac, of the male's display improve a female's ability to recall the location of that signal? But first I have to go out and catch some frogs, right? My team and I work out of Gamboa, Panama at the Smithsonian Tropical Research Institute, where we go out at night and collect pairs that are already in amplexus, like in the previous slide where the male is on top of the female in a little backpack position. After collecting them from the field, we bring them back to our lab where we dark adapt them for at least 1 hour to allow their eyes to be able to see more clearly and make a visual decision. As for our actual experimental conditions. So here we have our sound chamber where your view is if you're standing inside and looking down. So there's going to be 2 speakers placed 80 centimeters away from where the female is going to be held in a funnel. A wine chuck call is being played at both speakers, and a RoboFrog is only placed at one. The blue crescents coming off from the speakers represent the acoustic signal, and the red squiggly lines coming off of the RoboFrog represent the visual cue. After a presentation period of 2 minutes, we release the female from the funnel and allow her to make a choice. For our memory experiments, which I call the blind cloth test, everything's relatively the same. We have 2 speakers 80 centimeters away from the female, both of which are playing the wine chuck call and only one has a RoboFrog. We still have a presentation period of 2 or 3 minutes, but after that, I lower the blind cloths. The blind cloths are made from a black opaque material that wraps around the speakers and the RoboFrog 360 degrees so that there's no indication of which speaker the RoboFrog is at, except what's already in the female's memory. After we lower the blind cloths, we sometimes add a silent period of 5 seconds, and we may also add an acclimation period of 20 seconds where we continue to hold the female in the funnel after the cloths have been lowered for 20 seconds before releasing her. Depending on the treatment, after each period is over, we lift the funnel and allow the female to make a choice. Let me familiarize you with our graph before we go any further. So here we can see there's a dotted line at the 50% mark. So we're going to be using a binomial test, which then asks if females are choosing at random or not. So, for example, if this bar was at the 50% mark, we would then know that females are choosing the unimodal bare speaker at an equal rate as the multimodal speaker with the RoboFrog. If this bar was lower than 50%, then that would mean that females are actually choosing the call or the bare speaker more than the RoboFrog. But as we can see here with a sample size of 46, females chose the RoboFrog speaker significantly more than the bare speaker, which isn't surprising given what we know about their preferences. For this next experiment, I just wanted to see if there's anything weird or aversive about either of the blind cloths I use, which is represented by the little blind cloth off to the right there. And so with a sample size of 13, we almost have a perfect 50-50, so there wasn't anything weird about the cloths. For our first memory experiment, I first exposed the females to the RoboFrog and the speakers during a 2-minute presentation period, after which I lowered the blind cloths over both speakers. After lowering the cloths, I then gave them an acclimation period of 20 seconds, where I continued to hold the females under a funnel and the calls at both speakers continued to play. After the 20 seconds were up, I released her and allowed her to make a choice. But what was surprising here is that females weren't significantly able to remember where the RoboFrog male was after those 20 seconds. So my team and I kind of brainstormed for a little bit, and then we decided, well, sometimes there are pauses in male choruses, so what if we added in a silent period? And so, we did. For this next experiment, I did relatively the same thing. The females had a presentation period with the RoboFrog and the speakers playing, after which the blind cloths were lowered. But then I turned the calls off for 5 seconds, and immediately when I turned them back on, I released the female and allowed her to make a choice. And with this, we saw that females were actually able to remember where the RoboFrog was. But of course, someone might say, well, the only reason the female will remember during that treatment, not the first memory treatment, is because the first one had a 20-second delay after the cloths were lowered, and this one had only 5 seconds. So, we then combined both of our previous experiments into one, and we continued to see the same results. So, to reiterate, for this last experiment, we again had a presentation period with the RoboFrog, followed by the cloths being lowered, but as soon as the cloths were lowered, both speakers were turned off for 5 seconds. After those 5 seconds, the speakers continued playing the calls, and an acclimation period started where the female was held under the funnel for 20 seconds. After a combined 25-second delay since she was last exposed to the RoboFrog, the female was then released and allowed to make a choice. As we can see, the majority of females chose the speaker with the RoboFrog, which is super exciting given that the only difference between this last treatment and the first blind cloth treatment was a 5-second silent period. This all leads me to say that the visual component of the vocal sac was able to improve the ability of females to remember the location of a calling male. And when you look at it, it's super interesting that the silent period was actually the thing that induced her memory. You know, to a human, 25 seconds, let alone 5 seconds, doesn't sound very long at all, but for a female túngara frog, maybe that silent period is biologically relevant because these females are arriving to a lek and are constantly temporally updating as they evaluate potential mates. Females are probably making a choice in just a few minutes, and in an area that has a lot of predation risk, 5 seconds might actually mean a lot to her. And if I'm anthropomorphizing, a silent period might jolt the female into being like, oh, I have to pay attention. And again, remember that these frogs are super tiny, so if in one moment she might see one male but only hear another, if she takes a little hop to the right behind a dirt clod and can't see either male, maybe she's more willing to go to the male that she already knows where he is, instead of trying to find a new one. So, in the end, the visual cue of a male's multimodal display might help a female remember the location of individual callers as she makes her decision. I'd like to thank everyone involved in this project. Our collaborators Rachel Page and Michael Ryan, our fieldwork assistants Jorge Lopez and Leslie Barria. Of course, none of this would be possible without the people who, helped create and make the RoboFrog possible, Paul Clements and Golden Frog Scuba, and Topher Manitz for graphic design. I'd also like to thank the Smithsonian Tropical Research Institute for logistical support and the NSF for funding, as well as my home institution of Salisbury University. Thank you.